Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology for October and November 2022, the scientific article for Unit 5. So we're going to be looking at the potential questions as well as going through and breaking down all the article. Let's begin with the first part. Here they say, how do we perceive the world? So this is about sensation as well as formation of impulses and so on. So they say, one of the great mysteries facing humanity is the question of how we sense our environment. The mechanisms underlying our senses have triggered our curiosity for thousands of years. For example, how light is detected by the eyes, how sound waves affect our inner ears, and how different chemical compounds interact with receptors in our nose and mouth, generating smell and taste. We also have other ways to perceive the world around us. Imagine walking barefoot across a lawn on a hot summer's day. You can feel the heat of the sun and the caress of the wind and the individual blades of grass underneath your feet. This impression of temperature, touch, and movement are essential for our adaptation to the constantly changing surrounding. So paragraph 2 says, in the 17th century, the philosopher René Descartes envisioned threads connecting different parts of the skin with the brain. In this way, a foot touching an open flame would send a mechanical signal to the brain, as we're going to see in figure 1. And discoveries later revealed the existence of specialized sensory neurons that register changes in our environment. Joseph Ellinger and Herbert Gesser received the Nobel Prize in Physiology or Medicine in 1944 for their discovery of different types of sensory nerve fibers that react to distinctive stimuli. For example, in the response to painful and non-painful touch, we've seen that. Since then, it has been demonstrated that nerve cells are highly specialized for detecting and transducing different types of stimuli, allowing a perception of our surroundings. For example, our capacity to feel differences in the texture of surfaces throughout the fingertips or our ability to discern both pleasing, warmth, and painful heat. Paragraph 3. Prior to the discoveries of David Julius and Ardem Patapoutian, our understanding of how the nervous system senses and interprets our environment still contained a fundamental unsolved question: how are temperature and mechanical stimuli converted into electrical impulses in the nervous system? So let's move on. So this is what they were talking about. Figure 1, illustration depicting how the philosopher René Descartes imagined how heat sends mechanical signal to the brain. So this is what they sensed. Yes, sends impulses like that to the brain. That is the image he envisioned. Paragraph 4, the science heats up. In the latter part of the 1990s, David Julius at the University of California, San Francisco, USA, saw the ability for major advances by analyzing how the chemical compound capsaicin causes the burning sensation we feel when we come into contact with chili peppers. Capsaicin was already known to activate nerve cells causing pain sensation, but how this chemical actually exerted this function was an unsolved riddle. Julius and his co-workers created a library of millions of DNA fragments corresponding to genes that are expressed in the sensory neuron, which can react to pain, heat, and touch. Julius and colleagues hypothesized that the library would include a DNA fragment encoding for the protein capable of reacting to capsaicin. They expressed individual genes from this collection in cultured cells that normally do not react to capsaicin. After a laborious search, a single gene was identified that was able to make cells capsaicin sensitive. We're going to see in figure 2. The gene for the capsaicin sensing had been found. Further experiments revealed that the identified gene encoded a novel ion channel protein, and this newly discovered capsaicin receptor was later named TRPV1. When Julius investigated the protein's ability to respond to heat, he realized that he had discovered a heat-sensing receptor that is activated at temperatures perceived as painful, as we can see in figure 2. So let us continue. So here we can see this is the molecule got from chili pepper. It was transferred into the cells, as we can see, and then the experiment was carried out. So figure 2 is about David Julius using capsaicin from chili peppers to identify the TRPV1 an ion channel activated by painful heat. Additional related ion channels were identified, and we now understand how different temperatures can induce electrical signals in the nervous system. Paragraph 5. The discovery of TRPV1 was a major breakthrough leading the way to the unraveling of additional temperature sensing receptors. Independently of one another, both David Julius and Adam Paraputian used the chemical substance menthol to identify TRPM8, a receptor that was shown to be activated by cold. Additional ion channels related to TRPV1 and TRPM8 were identified and found to be activated by a range of different temperatures. Many laboratories pursued research programs to investigate the roles of these channels in thermal sensation by using genetically manipulated mice that lacked these newly discovered genes. David Julius's discovery of TRPV1 was the breakthrough that allowed us to understand how differences in temperature can induce electrical signals in the nervous system. Research under pressure. We'll look at paragraph 6. It says, while the mechanisms for temperature sensation were unfolding, it remained unclear how mechanical stimuli could be converted into our senses of touch and pressure. So here they say, researchers had previously found mechanical sensors in bacteria, but the mechanisms underlying touch in vertebrates remained unknown. Adam Paraputian, who worked with the Scripps Research in La Jolla, California, USA, wished to identify the elusive receptors that are activated by mechanical stimuli. Paragraph 7. Paraputian and his collaborators first identified a cell line that gave off a measurable electrical signal when individual cells were poked with a micropipette. It was assumed that the receptor activated by mechanical force is an ion channel, and in the next step, 72 candidate genes encoding possible receptors were identified. These genes were inactivated one by one to discover the gene responsible for mechanosensitivity in the studied cells. After an order search, Paraputian and his co-workers succeeded in identifying a single gene whose silencing rendered the cells sensitive to poking with a micropipette. A new and entirely unknown mechanosensitive ion channel had been discovered and was given the name piezo1 after the Greek word for pressure. Through its similarity to piezo1, so a second gene was discovered and named piezo2. Sensory neurons were found to express high levels of piezo2. Studies firmly established that piezo1 and piezo2 are ion channels that are directly activated by the exertion of pressure on cell membranes, as we can see in figure 3. Let's continue. So here we can see figure 3. We have the gene silencing that was carried out, and then the experiment studied the effect of pressure, and that in order to observe which genes were responsible, as we can see. So here they say, figure 3, Paraputian used cultured mechanosensitive cells to identify an ion channel activated by mechanical force. After painstaking work, piezo1 was identified. Based on its similarity to piezo1, a second ion channel was found as well. So that was called piezo2. Paragraph 8 says, the breakthrough by Paraputian led to a series of papers from his and other groups demonstrating that piezo2 ion channel is essential for the sense of touch. Moreover, piezo2 was shown to play a key role in the critically important sensing of body position and motion known as proprioception. In further work, piezo1 and piezo2 channels have been shown to regulate additional important physiological processes, including blood pressure, respiration, and urinary bladder control. Paragraph 9. The groundbreaking discovery of the TRPV1, the TRPM8, and piezo channels by this year's Nobel laureates have allowed us to understand how heat, cold, and mechanical force can initiate the nerve impulses that allow us to perceive and adapt to the world around us. The TRP channels are central for our ability to perceive temperature. The piezo2 channel endows us with a sense of touch and the ability to feel the position and movement of our body parts. The TRP and piezo channels also contribute to numerous additional physiological functions that depend on sensing temperature or mechanical stimuli. Intensive ongoing research originating from this year's Nobel Prize awarded discoveries focuses on elucidating their functions in a variety of physiological processes And this knowledge is being used to develop treatments for a wide range of disease conditions, including chronic pain, as we can see in figure 4. Involving the TRPV1, we see temperature or heat pain. We see core body temperature, inflammatory pain, neuropathic pain, visceral pain, and protective reflexes. Involving piezo2, we see touch as well as proprioception. We see mechanical pain, urination, respiration, blood pressure, and skeletal remodeling. So here they say figure 4 is the seminal discovery by this year's Nobel laureates have explained how heat, cold, and touch can initiate signals in our nervous system. The identified ion channels are important for many physiological processes and disease conditions. In order to break down the questions, I'll begin with paragraph 1. In paragraph 1, I saw they could ask a general question about response to our environment. It could include talking about impulse generation in response to any change, the brain's involvement, as well as negative feedback mechanisms. So this could be a very broad question, and it could come on any of the paragraphs. The next segment I saw is how light is detected by the eyes. So in this case, since you talked about the pupil reflex and so on in your textbook, so I thought they could ask about the pupil reflex involving the scapular muscles, longitudinal muscles of the iris, and how their contraction or relaxation can lead to pupil dilation and constriction. Then next I thought about, since we're still talking about the eye, there is rhodopsin. So in this case, they could talk about the generator potential. So think about rhodopsin, how when rhodopsin is struck by light, it could go through a conformation change. Think about that cascade of reactions that happen after that and how they lead to formation of a generator potential. The third part I saw is coming from this part here where they say different chemical compounds interact with receptors. So here it could be anything from within the body or from the external surrounding of the body. So I said how receptors on the cell function by binding with chemicals to cause change inside the cell. This could be any chemical, including even hormones. So it could be chemicals or hormones that work as transcription factors. Please revisit your textbook, page 214 to 215. Another thing could be, since receptors can be protein molecules, they could also ask about protein synthesis. They could ask about the primary structure of a protein, secondary structure, tertiary structure, as well as how they fold. So this could be very broad, different chemical compounds interacting with receptors. The key thing here is you have to remember they bind. Sometimes they cause their change inside the cell from the outside, or sometimes they bind, they enter the cell, and they cause their change from within the cell. So those two things have to be taken into account, and you have to think about that page. Please revisit that and understand that fully. From paragraph 2, because they were talking about specialized sensory neurons that register changes in our environment, I thought about how since certain neurons are connected to receptor cells, these will respond or react to distinctive stimuli, and that is going to lead to formation of an impulse that is transferred or transported through the sensory neurons. So I thought they would talk about sensory neurons and the ability to respond to the different types of distinctive stimuli. They are connected to specific receptors that are going to detect the change, convert that change into an impulse or an electric signal that is going to be transported through the sensory neuron to the central nervous system. So here I said you have to remember that this stimulus will change the membrane potential of the receptor cell. So there is a receptor cell, its potential is going to change. In the process of that change, an action potential is going to be created within the sensory neuron, and this will take the electrical signal to the central nervous system. They could also ask a question about a reflex action. This is not direct, but I just thought about it. So you have to revisit what is a reflex action and how does it occur. In paragraph 3, this is they ask how are temperature and mechanical stimuli converted into electrical impulses in the nervous system. Exactly the same as we see here. So you can see paragraph 2 and paragraph 3, it is leading to an inclination of how an impulse is generated in a sensory neuron. So you need to know how receptor cells are changed when a stimulus arrives in order for the electric signal to be created and in order for that electrical signal to be transferred to the sensory neuron to be carried to the central nervous system. So you need to know that. Now, I also thought about some bonus questions. Because we are talking about transfer of an impulse through the neurons, they could talk about the myelination and speed of an impulse. You have to know neurons that have myelin around them carry an impulse at a faster rate than neurons that are not myelinated. So they could ask about myelination and impulse transmission speed. They could talk about propagation of an action potential across the membrane within the axon. They could talk about the synapse, neurotransmitters, and impulse transfer across the synapse. They could talk about, they could ask you to link disease of the nervous system and why certain infected people might be slow or not responsive to certain chemicals or certain environmental changes. That is involving maybe the nervous system, muscles, and so on. They could also ask about the nervous system and negative feedback involving any change in the environment. This change could be maybe due to temperature. For paragraph 4, because they talked about that they created a library of millions of DNA fragments corresponding to genes that are expressed in the sensory neuron, which can react to pain, heat, and touch. Here they could connect a question about polymerase chain reaction. Also, because they talked about the idea that they expressed individual genes from this collection in cultured cells. So we need to talk about gene expression and we need to talk about culturing cells. So I thought they could ask a question about the transcription, post-transcription modification, including splicing, translation, post-translation modification, folding of proteins into our secondary structure, tertiary structure, quaternary structures, and so on. They could also include cell culturing techniques because we have this. So how were they able to express the individual genes? We have to talk about epigenetics. Moving to paragraph 5. In paragraph 5, we saw they are using genetically manipulated mice that lacked newly discovered genes. And they also talk about how differences in temperature can induce electrical signals in the nervous system. So here I thought they could talk about genetic manipulation as well as modification. So a question on how genetic modification is done could be asked. Please revisit your A2 biology book, page 292 to 295, in order to ensure that you understand everything about production of recombinant DNA. They could also ask about restriction enzymes, ligases. They could talk about vectors and gene delivery into the target. They could also talk about methods of identifying that the organism has been transformed. The example in the textbook is replica plating. Also, they could ask about silencing of genes. You have silencing of genes within this page here. It's about page 294 to 95. So you have to think about that, how gene silencing is carried out to create knockout organism, because here they say they used manipulated mice that lacked these newly discovered genes. Maybe there was gene silencing, or they created knockout organisms that they used for this experiment. So you have to think about that. Paragraph 6, here they talk about the mechanical stimuli could be converted into our senses of touch and pressure. So this is about formation of an action potential or formation of a nerve impulse from the stimulus. Also, in paragraph 7, they're talking about, they said it was assumed that the receptor activated by mechanical force is an ion channel. So please focus on how ion channels are involved in generation of impulses as well as action potentials. So remember, ion channels they open or close to allow movement of specific ions in different directions in order to cause a potential difference so that electrical signals can be generated in order for impulses to go through the neurons into the central nervous system. Also here they talk about genes encoding possible receptors. So since receptors are protein in nature, they could talk about how information moves from DNA in order for protein to be made. So that is about protein synthesis. Then the next segment talks about these genes were inactivated one by one. Inactivation of genes is silencing of genes. So you need to revisit your textbook like we saw already in paragraph 5 and think about how genes are silenced. And lastly here they say whose silencing rendered the cells insensitive to poking with a micropipette. I want to take you back to paragraph 5. You have to read about how gene silencing leads to formation of knockout organisms. So continuing. Paragraph 8, they say, moreover, piezo2 was shown to play a key role in the critically important sensing of body position, the motion known as proprioception. So here, I thought they could include the cerebellum, how impulses from these regions are sent to the cerebellum, which is the region of the brain concerned with motor movement, balance, as well as the ability to determine limb position. So you could think about this. This is as well as impulse transmission. So most of these questions are going to be about how a stimulus is converted into an impulse, how the impulse is carried through the neurons to the central nervous system, and how a response is going to be seen. Lastly, here they say, in further work, piezo 1 and piezo 2 channels have been shown to regulate additional important physiological processes. So these two regulate blood pressure, respiration, as well as urinary bladder control. It means they could ask you about questions of nervous control of blood pressure involving sympathetic, parasympathetic, sinoarterial node, atrioventricular node, and so on. They could also ask nervous control of respiration. Here is, remember, cells that respiring, they could have a higher rate of respiration or a lower rate of respiration. So stimulation of specific cells could cause them to respire more. And also they could ask nervous control and the bladder. Remember, the bladder has some specific muscles. Those muscles could be stimulated to either contract or open up in order to let the urine to be passed out. So this could be muscular contraction, the sphincter muscles, and so on. In paragraph 9, here I read, they say, these people who won the Nobel Prize helped them to understand how heat, cold, and mechanical force can initiate the nerve impulse. So it means this is the stimulation that causes specific cells to be responsive in order to change so that an electric impulse is generated. So we have to look at how a nervous impulse is generated due to the changes that could occur in the membrane of the specific receptor cells. Also, they say this knowledge is being used to develop treatments for a wide range of disease conditions, including chronic pain. Now, if we know the information or we know the genes that are coding for the specific receptors or for the specific channels, we could find a way of modifying, if possible, or find a way of causing blockage, or it could be silencing, any method that could be applicable. So I say, potential treatments for chronic pain and other conditions could be, since the genes coding for these channels are known, it is possible, or there is a possibility of silencing these genes. Also, there is a possibility for the formulation of medication that could block the ion channels so that impulse generation in case of chronic pain is going to be inhibited. So if the ion channels are blocked, ions cannot move in and out, so the impulse is not going to be generated, so these people are not going to feel unnecessary pain in those areas. So this brings us to the end of this scientific article. I hope it helps you a lot. Please do not forget to subscribe to our channel. See you in the next video. Bye-bye.